Kingdom fungi. The kingdom fungi includes heterotrophic organisms. In morphology and habitat, they show a great diversity. It is because of fungi that the bread develops a mold or the orange rots. The mushrooms that we eat and toadstools belong to fungi. When white spots are seen on mustard leaves, then that is due to the parasitic fungus. Yeast is a unicellular fungi which is used to make bread and beer. Some fungi cause diseases in animals and plants. Example Puccinia, which causes wheat rust. Antibiotics are manufactured from some fungi. Example Penicillium. Fungi are present everywhere in soil, water, air, and on plants and animals. Fungi grow in humid and warm places. Food is kept in the refrigerator to prevent food from going bad due to bacterial or fungal infections. Fungi are filamentous, but the yeasts are unicellular fungi. The bodies of fungi are long thread-like structure called the hyphae, and a network of hyphae is called mycelium. Some hyphae have multinucleated cytoplasm as they are continuous tubes without any cross walls, and such hyphae are called cenocytic hyphae. The other fungal hyphae have septae or cross walls in their hyphae. Chitin and polysaccharides form the cell wall of fungi. Most fungi are called saprophytes as they absorb organic matter from dead plants and animals and hence are heterotrophs. Those organisms that depend and live on living animals and plants are called parasites. Some fungi live in association with algae together called as lichens, and some other fungi live in the roots of higher plants as mycorrhiza. Vegetative means of reproduction is seen in fungi such as fragmentation, fission, and budding. Asexual and sexual reproduction are also seen in fungi. Zoospores, sporangiospores, or conidia are the spores produced by fungi during asexual reproduction, and oospores, ascospores, and basidiospores are produced during sexual reproduction. These spores produced during reproduction by the fungi are borne in structures called fruiting bodies. Three steps are involved in the sexual cycle. First, plasmogamy. In this, the protoplasms of the two motile or non-motile gametes fuse. Second, karyogamy. In this, two nuclei of the two gametes fuse. Third, the zygote formed after fusion undergoes meiosis resulting in haploid spores. During sexual reproduction, fusion takes place between two haploid compatible mating types of hyphae. In some fungi, the diploid cells are formed immediately after the fusion of two haploid cells. However, in Ascomycetes and Basidiomycetes, a dikaryotic stage is formed and is called a dikaryon, and the phase is called dikaryophase. The parental nuclei fuse later and diploid cells are formed. The fungi then develops fruiting bodies. In these fruiting bodies, reduction division occurs and haploid spores are formed. The basis for dividing of the kingdom into different classes involves morphology of the mycelium, mode of spore formation, and fruiting bodies. Phycomycetes. Fungi included under Phycomycetes are found growing in aquatic habitats. They are also found on decaying wood in damp and moist places, and some are found growing on plants as obligate parasites. The mycelium in Phycomycetes is aseptate and cenocytic. Asexual reproduction takes place by the formation of zoospores which are motile or by the formation of aplanospores which are non-motile. The spores formed are produced by sporangium endogenously. Phycomycetes produce zygospores and they are formed by the fusion of two gametes. The gametes formed are isogamous and or anisogamous or oogamous. Some common examples of fungi included under Phycomycetes are Rhizopus, the bread mold, albugo, the parasitic fungi that grows on mustard, and mucor. Ascomycetes. Ascomycetes are commonly known as sac fungi. The sac fungi are unicellular as in yeast, Saccharomyces, or multicellular as in Penicillium. They are saprophytic and decomposers. Some of them are parasitic or coprophilous, growing on dung. The mycelium of Ascomycetes is septate and branched. The asexual spores called conidia are produced exogenously. Conidia are borne on special mycelium called conidiophores. These conidia later on germination develop into mycelium. Ascospores are produced during sexual reproduction. These ascospores are produced endogenously in a sac-like structure called asci. These asci are produced in fruiting bodies called ascocarps, example Neurospora, claviceps, and aspergillus. The fungi Neurospora is often used in biochemical and genetic work. Some of the members of Ascomycetes are edible and considered as delicacies, example morels and buffles. Basidiomycetes. Some common examples of basidiomycetes are puffballs, bracket fungi, or mushrooms. Basidiomycetes grow on tree stumps, logs, and soil. Some live as parasites in the living plant bodies like rusts and smuts. The mycelium in basidiomycetes is septate and branched. Vegetative reproduction is by fragmentation and asexual spores are usually not found. The sex organs in basidiomycetes are absent, but two vegetative or somatic cells of different strains or genotypes fuse to give rise to a dikaryotic structure which finally gives rise to basidium. This is known as plasmogamy. Four basidiospores are formed in the basidium by karyogamy and meiosis. The basidiospores are produced exogenously on the basidium. The basidia thus formed are arranged in basidiocarps, which are the fruiting bodies. Some common basidiomycete members are Puccinia, rust fungus, Astilago, smut, and Agaricus, mushroom. Deuteromycetes. Deuteromycetes are known as imperfect fungi because only the vegetative or asexual mode of reproduction are known. When the sexual mode of reproduction were discovered in these fungi, they were moved into their respective classes. It can also be possible that the fungi that showed vegetative and asexual mode of reproduction were placed under Deuteromycetes and the sexual stage were placed under a different class. Later, when the linkages were established and the fungi were correctly identified, they were removed from Deuteromycetes. When the sexual stages of members of Deuteromycetes were found, they were often placed under Ascomycetes and Basidiomycetes. So the Deuteromycetes reproduce only by asexual mode of reproduction by producing spores known as conidia. When observed under microscope, their mycelium was septate and branched. Some of the members are saprophytes and some are parasites. However, a large number of deuteromycetes are decomposers and help in mineral cycling, example Trichoderma, Colitotrichum, and Alternaria. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click the bell notification that would let you know when a new video is uploaded. Thank you so much for watching.